We are live.
Good afternoon, friends, and a warm welcome to everyone. I, Dr. Sukhjeet Kaur Gujral, coordinator of Guru Nanak Institute of Research and Development of Guru Nanak Khalsa College, Mumbai, am delighted to welcome you all to the webinar titled Development of Biologics, Gene Therapy, and Scientific Writing. Teaching faculty, professors, research scholars, industry personnel, and students from many states of India and some international researchers are participating in this webinar. I'm extremely thankful to our college principal, Dr. Kiran Mangaonkar, the R&D department, the HOD, Dr. Gagan Jyot, and the technical team for their constant support and encouragement to conduct this webinar. I would like to give you a small highlight of our college. Guru Nanak Khalsa College of Arts, Science and Commerce is an autonomous college under Shiromani Gurdwara Prabandhak Committee. Next slide. It has a heritage structure, as you can see, and it was established in the year 1934. It has students right from 11th standard up to PhD studying in this college. It has got the Best College Award from the University of Mumbai. It has got the College for Potential for Excellence Award. It has been accredited by NAC with an A grade and with a score of 3.54. Khalsa College is scientific, industrial, and research organization that is zero recognized. It has a reputation for excellence in sports also. Guru Nanak Institute for Research and Development offers MSc and PhD program. It also has 4 MSc programs, namely MSc in Bioanalytical Sciences, MSc in Bioinformatics, MSc in Nutraceuticals, and 5 years integrated course in Bioanalytical Sciences. I think ours may be the only college in all of India which has these 4 innovative UGC MSc programs, which are job-oriented. Khalsa College also has the National Facility for Biopharmaceuticals for advanced research in molecular biology. We have received so many grants from various granting agencies in our college. Guru Nanak Institute of Research and Development was established in the year 2005 as the research wing of the college. It has a state-of-art facility with many sophisticated analytical instruments. Here we maintain a very conducive and stimulating environment so that the trained research scholars are ready to serve the growing need of academia, research, and industry at both national and international level. The research facilities that we provide to the students include a microbiology lab, bioinformatics tools, lab and softwares, nutraceutical product development and testing labs, analytical method development on the instruments like HPLC, GC, and HPTLC, molecular biology lab, fermentation technology, bioprocess technologies available. We have labs for sensory evaluation of food, biotechnology related services, and we definitely take our projects in a very big way. The research areas are very diversified. Students can opt for the topics mainly in health and nutrition, environmental biotechnology, food microbiology, agriculture, phytochemistry, medicine inclusive of cosmeceuticals and diagnostics, and separation technology. We have many research activities in GNIRD. We conduct MSc and PhD program in interdisciplinary science, namely bioanalytical sciences. We organize and conduct training programs, workshops, and refresher courses. We have alliances with many industries for joint collaborative research work. We very aggressively offer consultancy to industries, college, teachers, and students for conducting research. And we are taking up research projects, short-term courses, and training programs in a big way. Since its establishment, we have got 11 research guides only associated with GNIRD. Otherwise, in the college, we have other 35 research guides. 46 students have been already awarded MSc PhD degree, and currently 30 students are pursuing their MSc and PhD program. More than 100 publications are there in peer-reviewed journals, and many sequences have been submitted to NCBI and GenBack. We encourage our students and research scholars to participate in workshops and undertake certificate courses so that they develop skills and training to have better job opportunities. These include certificate course in cosmetology, in IPR issues, SPSS, Linux, in sensory evaluation of food, and a very intensive workshop on chromatographic and spectroscopic techniques. Thank you. Investment in knowledge pays the best interest, as said by Benjamin Franklin. And in this lockdown, let us invest in an academic delight by a presentation and talk by Dr. Chirmule. It's an opportunity to have a dynamic technocrat and renowned researcher, Dr. Chirmule, with us today for this particular webinar. Friends, it is my proud privilege to introduce him to you. Dr. Narendra Chirmule is the co-founder and CEO of Symphony Tech Biologics, a data analytics company focused on engineering solutions to biology. As a former head of R&D at Biocon Bangalore and having leadership positions in Amgen and Merck, he has contributed to clinical development of vaccines and biopharmaceuticals. He did a PhD on development of a leprosy vaccine from the Cancer Research Institute, Mumbai, followed by postdoctoral studies on pathogenesis of AIDS from Cornell University Medical College, New York. Sir is teaching and doing research as assistant professor in gene therapy at the University of Pennsylvania. Dr. Chirmule is also on the NIH Advisory Committee for HIV vaccines. This was just a brief introduction of Sir. I'm sure all of you are waiting eagerly to hear to this dynamic speaker. I welcome Sir, and I'm very grateful for him joining us this webinar. Sir, the stage is all yours. Thank you so much.
Thank you very much. Thank you. I will present now. Okay. Can you see my slides? Let me know if you can see my slides. Okay. Can someone just confirm, because I cannot see you. I don't know if you can see my slides or not. So
yes, sir, we can.
Sir.
You can. Okay, excellent. Thank you. Okay. Thank you so much for inviting me to this presentation, and it is my pleasure to talk to you a little bit about my journey on gene therapy, and I'll touch a little bit on scientific writing. But quick background about myself. My name is Narendra Chirmule, again, as Dr. mentioned. I've done my PhD from Bombay University. Actually, I went to Junjunwala College, which is in Ghatkopar. I used to live in Matunga, which is very close to Khalsa College, and several of my friends went to Khalsa College. And Mumbai, I love Mumbai. And I did my PhD at Cancer Research Institute in Parel. After which I went to the US to do my postdoctoral fellowship in North Shore University, Cornell University in New York City, where I worked on HIV pathogenesis. After which I went to University of Pennsylvania and I did research on gene therapy. That was my first exposure in the early 1990s. And then I went from academia to industry. I joined Merck, which is the vaccine company. I worked on development of many vaccines there. After which I went to Amgen, which is in Thousand Oaks, California, where I did some work on development of biologics. But 5 years ago, I came back to India after spending 30 years in the US, and I was head of R&D in Biocon, and I worked closely with the team there. And we were fortunate to develop several biologics and got them approved in the US. So that was sort of my experience in my career. In my career, I talk about inflection points, and so I guess my key inflection points was one of the inflection points was when I was going to do my PhD. जब मैं PhD करने वाला था Cancer Research Institute में, तो मेरे को एक मौका मिला था कि to join a company called Hext उस time पे to do marketing. So after my master's, I had that choice, and then I don't know what made me make that decision, but I decided that I would do my PhD rather than marketing. So that inflection point for me was that decision point of going towards PhD. And then, of course, another inflection point was to return to India after 30 years in the US. And I can honestly tell you, after coming back to India, I stay both in India and the US. In fact, I'm in Philadelphia right now. I really have got a lot of experience in working and enjoy my job both in various parts of the world. And personally, I've learned to play the tabla for many years, and I play tabla quite a bit. But in the last couple of years, I've been learning to play the flute. So that's sort of my overall background. So let's get started with gene therapy. The reason I put this slide up is because I think the fields of biology today are merging. And in Khalsa College, I heard a lot about from your professors about how different fields are coming together. And in the field of gene therapy, it is really the merging of many different fields of medicine, all the pathological diseases, understanding of molecular biology, biochemistry, bioinformatics, all of that coming together, which makes all of this happen. But the other thing that also we forget about is, as in the field of science, is that arts and commerce is also important in our overall picture. So we need to understand how everything we know comes together. So that's part of how we have to follow our dream. When we talk about hemophilia, or actually when you talk about genetic diseases, I'm sure many of you have known or know someone in your friend circle who has some kind of genetic disease. So although each genetic disease is rare, collectively, there are a lot of us who have various genetic diseases. So Dr. Murali, who's shown here in this picture, and I have been friends for 40 years. He lives in Chicago. He has a factor VIII deletion in his gene. and from which he gets severe bleeding. In fact, when he just climbs a flight of stairs, he will have internal bleeding, right? But in addition to the bleeding, he has severe joint damage to his knees, elbows, and shoulders. So he's got a lot of arthritis. And a couple of years ago, just 2 or 3 years ago, 2 years ago, his kidneys were having a problem. So he had a kidney transplant from his brother. And he now is thinking of a knee replacement surgery. And you can imagine the kind of life he has been leading. But personality-wise, he's the person with the most positive attitude that I ever know. He's such an amazing individual. I talk to him a lot. So, the hemophilia, we talk about all these diseases, but the diseases actually are people. So, it is very important to know about the wholesome aspect of the disease. DNA, I'm not going to spend too much time on the history of molecular biology, except to say that in 2010, when the Human Genome Project came alive, both from the industry as well as from academia, the possibilities of what could happen changed our lives. So this is a picture of a cell where you can see the DNA being expressed in the cell, converted to RNA. RNA then through the ribosome is converted to protein. Protein goes through this fantastic post-translational modification. And so we have all of physics, chemistry, biology, and dare I say quantum physics involved in the process of molecular biology. When the genome was sequenced, it was only one genome that we understood. Over the last several decades, we know that although the genome is very, very similar, human genome is very similar to many other species that we know of, it really is the basis of the diversity of the human population. So genomic sequences really tells us from a molecular level all the differences that we have. Gene therapy really had started many, many years ago, even before the Human Genome Project started. And this is a picture which really demonstrates the power of gene therapy and what we could do potentially. These are animals, green fluorescent protein. This is a gene which is called a green fluorescent protein, which is taken from one of the flies, which fluoresces. That protein which fluoresces under UV light is the gene for that is inserted into the embryonic stem cell of such animals. And when the animals form, the animals start expressing the green fluorescent protein. So when you shine light on them, on these mice, for example, the mice turn green because they're expressing a foreign gene. Now, this process of looking at GFP is called reporter gene expression. So this is the power of gene therapy. Historically, the main events that are listed here really are the journey of gene therapy, but which really highlighted by the fact that there were today's success, which is coming in 2019 or 20, the approval of Luxturna and the drug from Novartis, has taken a long time to develop and a lot of challenges. So I'm going to talk about a few topics in the course of this lecture about this journey of gene therapy. There are around 30,000 genes in our human, in the humans, but interestingly, only 200 or 300 or odd genes are genes that are targeted to drugs that have ever been made. So that number is astronomically small. So let me say that again. Of the 30,000 genes, only about 300 genes against which we have targets, very clear targets on which we have drugs today for. So imagine the potential of how many more drugs we can develop if we understand the human genome much better. more than 1,000 clinical trials going on in various phases of clinical trials on gene therapy itself. From a historical perspective, there are lots of things that are happening in gene therapy that are listed in this slide, but I will talk about the ones that are marked in blue. I'll talk a little bit about cystic fibrosis. I'll talk a little bit about this disease with ornithine transcarbamylase deficiency. I'll talk a little bit about retinal disease, and I'll end my talk with CAR T cell therapy, which is the current therapy. So these are the 4 topics I will cover during my presentation. And they have a reason for me to choose these molecules. So let's start historically in 1996. What is cystic fibrosis? So cystic fibrosis is caused due to a mutation in a gene called cystic fibrosis transmembrane receptor. So this gene is mutated. And this gene is expressed in the epithelial cells of the lung. And when this gene is absent, the transport of chloride ions through the CFTR is defective. You can see on the right, if you don't have the transmembrane domain of this receptor, the chloride ions cannot be efficiently transferred. The manifestation of this disease, the clinical manifestation of this disease, is due to reduced viscous secretions. So mucus is not being able to be secreted due to this loss of pH. Yeah. So for many years, it was an enigma as to why would a chloride channel result in higher mutations and higher viscosity, and then ultimately the patients die of a bacterial infection. So why would a bacterial infection be impacted by a gene in the chloride channel? So a very interesting experiment was done where you take the lung airway of an uninfected person. This is the epithelial cell, which has the chloride channel and the mucus proteins. And then you look at the cells, and then you look for this infection with Pseudomonas aeruginosa. Right? In the normal airway, there is no infection. And in the absence of the chloride channel, or defective chloride channel, it affects the amount of mucus, and this Pseudomonas infection, it becomes very rampant. Defensins are a particular kind of protein that are secreted by the epithelial cells. They are called antimicrobial peptides, and they are present, they are very, very conserved. So it turns out that the human defensins are misfolded in the epithelial tissue of the cystic fibrosis lung. And so therefore, when the chloride channels are blocked, the defensin proteins in the epithelial cells are not folded correctly, and therefore they cannot kill the Pseudomonas bacteria. And so the chloride channel, therefore, by changing the pH, loses the ability, induces the loss of ability of the epithelial cells to kill pathogens. So such a beautiful analysis of this pathogenesis of cystic fibrosis was demonstrated using the concepts of gene therapy. So when you think about gene therapy, you have to think about taking a piece of DNA and inserting it into humans. That's therapy. But DNA naturally exists inside the cell. So if you take a piece of DNA and you want to insert it, put it inside, you need a transport vehicle, which is in this case called a vector. So you put the gene that you're interested in and package it into a viral vector, those listed here, or a non-viral vector, like liposomes or naked DNA, and you have to be able to insert it. So viral vectors are very important for understanding how to transport DNA molecules into the cell. So we move to 1999. Adenoviral vectors. So adenovirus is a virus that causes the common cold. They are very good as vectors because they are easy to grow in culture. They infect non-dividing cells. and they are very well understood from a molecular biology perspective. So if we take an adenovirus and expresses the cystic fibrosis transmembrane channel that I just showed you, if that gene is expressed in the adenovirus and you infect the patient's lung, you can see that the patient's lung will now start expressing the cystic fibrosis gene. And therefore, you could potentially cure cystic fibrosis in theory. Similarly, you can also take ornithine transcarbamylase gene and treat that disease. So what is OTC deficiency? OTC is a X-linked gene which is responsible, the enzyme is responsible for a urea cycle disorder. Deficiency involves a series of biochemical steps in the liver in which nitrogen, which is a component of protein, is removed from the blood and converted to urea. The ammonia reaches the brain through the blood, where it may cause irreversible damage or death. So this is what OTC does. It causes accumulation of ammonia and causes death in little boys because it's an X-linked gene. So why choose OTC, such a complex disease, a rare disease, to treat gene therapy? Because it is a single gene defect. From many animal studies, we know that just the mutation of this gene causes this deficiency. It is X-linked. There are the females who are heterozygous have milder form of the disease. So we can also have, it's not a yes or no phenomenon. You can get milder diseases and treat women to see whether you can improve their deficiency. It is localized to the liver, and the mouse model of OTC deficiency was corrected. So that's why OTC gene makes a lot of sense to study gene therapy concepts. So in University of Pennsylvania, we did a clinical trial where we took the vector, which was the adenovirus vector, which contained the OTC gene. It was injected through the intraportal route, which is directly into the liver. And there were various doses given to each cohort of patients, from 2 × 10⁹ to 6 × 10⁻11. The results were the first 18 patients tolerated the therapy, but there was no significant expression of OTC gene in their liver. The 19th patient died after 5 days after vector administration. The trial was stopped and an intense investigation pursued. OTC-19, as he was called, was in a coma by the second evening after receiving the treatment that subsequently led to DIC, which is disseminated intravascular coagulopathy, which is the chronic inflammation response. No meaningful recovery was likely, so life support was withdrawn on day 5. Postmortem of this patient was done, and there was an unexplained insult to the bone marrow, hemorrhage and necrosis of the liver. Lungs were intensely inflamed. Kidneys were relatively spared. Severe anoxic result in the brain with encephalopathy and the infarcted spleen. Dr. James Wilson was in charge of that clinical trial at that time. And we took a lot of work, we did a lot of work at that time to understand why this patient died due to gene therapy. And the summary was that adenoviral vectors induce, although they are very good in transducing genes, they induce a lot of inflammation. And so therefore, it was not the ideal vector. Over the next decade, Jim did an amazing amount of work, and he discovered a new virus. He and a lot of other people in the field also, but his group primarily discovered another virus, which is called adeno-associated virus, and started developing the adeno-associated virus as a tool for gene therapy. So now we move on to 2007. Adeno-associated viral vectors were developed, and there were many these viruses isolated from many different sources, especially from animals, and looking at their expression in the liver, the ability of these viruses to express in the liver, in the lung, in the muscle, in the heart. So different viral vectors are able to express differently in different tissues. This was very good at expressing GFP in the muscle. You will see the muscles light up green. If you put AAV factor 9, which is one of the hemophilia diseases, and you put factor 9 into the AAV vector, GFP gene, which is the beta galactosidase gene, but if you treat animals who are deficient in factor 9, you can restore their hemophilia condition. If you now put AAV GFP, which is again green fluorescent protein, in the AAV and inject it into the eye, into the primate eye, this is the retina of the eye, and inject this vector, you can see that the AAV taking the GFP gene is expressed beautifully in the lining of the cells in the retina. So if there was a deficiency of the eye, you can actually transport a gene into the eye. That is what Gene Bennett did in University of Pennsylvania. There is a gene called RPE, which is a deficiency, the genetic mutation causes blindness. It causes slow retinal degeneration. And there's a genetic disorder in a dog of this disease, and the dog becomes blind. And this dog was treated with the AAV expressing RPE65, and this dog would actually start seeing. So that was one of the first demonstration that gene therapy could cure a disease. Since then, in 2019, Spark Therapeutics developed the gene, took that RPE65 and did a clinical trial. And now this drug is approved and is available for treatment of this kind of a very rare disease. And this eye disease can be treated with gene therapy. The drug is called Luxturna. Fast forward to 2019, current day. Lot of different viral vectors, a lot of people are working in the field of gene therapy now. It has transformed how we do gene therapy. Luxturna, I told you, is approved. This drug, Imlogic, which is from BioVex, a subsidiary of Amgen, treats cancers. Kymriah, which is, I will talk a little bit more about this. This is the CAR T-cell therapy. Yescarta and Zolgensma, which is the drug for a very unique kind of disease related to muscle disorder, has been approved. Gene Bennett, there's a beautiful YouTube video. When you have a chance, you can take a look at it. It demonstrates how this gene actually cures blindness by providing the normal gene to the patients who have a defect. And Kathy Hai, who has spent her entire career in studying hemophilia, is very close to developing clinical trials for factor 8 and factor 9 gene therapy. So patients with hemophilia, like my friend Murali, will get the benefit of being cured of hemophilia in the future. So I think it's very exciting times in the field of gene therapy where such therapies there'll be a cure for genetic diseases. And finally, I'll come back to Jim Wilson, because I think he is a pioneer in the field of gene therapy and my mentor, and he has done so much work in this field. So let's move on to another topic in gene therapy, which is more current. It is the use of gene therapy in cancer. In the last decade, activation of the immune system to kill tumors has become a very, very important tool in the arsenal of killing cancer, in addition to radiation, chemotherapy, and surgery. A little bit before we go into the treatment of cancer, about activation of T cells. When T cells, which immune killer T cells, for example, when they recognize antigen, they get activated. And when they get activated, they bind to these receptors on CD3 molecules, which are called CD3 zeta, and they bind to this, and they activate certain enzymes, which then activate various signal transduction domains, which then finally result in activation of the cell, which then results in their ability to kill their targets. Now, these are killer T cells that I just showed you. They recognize through the T cell receptor. And upon that activation, they release these toxins, and these cytotoxins are used to kill cells. And in this case, it's going to be kill cancer cells. And this is an electron micrograph of a T cell tumor cell. So what this T cell has done, and there's a whole group of people who are doing this work right now, what they've done a very interesting experiment where they have taken that intracytoplasmic tail that I showed you of CD3 zeta, which is the signaling domain of that T cell receptor, and they've linked it artificially to an ectodomain, which is an external portion of the protein, which contains the variable and light chain of the antibody molecule that recognizes the tumor. So it's a chimera between an antibody molecule, which is on the top of the cell membrane. It has a transmembrane portion, and it is linked to the cytoplasmic tail of a T cell receptor. And this portion transmits the signal, and therefore the CAR gene, which is the chimeric antigen receptor gene, is taken and put into a viral vector, in this case a lentiviral vector, and this vector is used to infect cells from a patient. And this kind of work is generally done in a hospital setting, where you have to grow the cells in a very, very clean facility. And you can see how the Tyvek people are wearing these Tyvek gowns and doing this analysis. And you can see this bag over here, which contains all the lymphocytes from the patient, which has been then infected with this CAR T expressing lentiviral vector. So the life cycle of this process is that the patient comes into the hospital, a cancer patient comes into the hospital, their cells are removed from their body by taking their blood. These cells are grown in culture for about 2 weeks, and they are then infected with the lentiviral vector expressing the CAR antigen that I showed you, the chimeric antigen receptor I showed you. So these T cells will now express an antibody which can recognize their tumor, and in the cytoplasmic tail they have the killer machinery, so that when these cells are put back into the patient, these cells go and kill the tumor in the patient. So one of the diseases that was first tried was acute lymphocytic leukemia. The prognosis for this disease is very bad. Generally, patients survive after getting into this very late stage of this disease. Patients survive for only weeks, not even years. But when 52 subjects were treated with this CAR T cell therapy, 83% of them had a very, very strong response within 3 months. So 83% of these people who were going to die had a very strong response. And overall survival, which is shown here on the right, 89% of them survived for 6 months, and 79% of them survived for 1 year. And now there are many cases where there are a lot of people who are being treated with this disease, with this treatment, who are surviving for a longer and longer time. One of the pioneers of this field is Carl June. He works in University of Pennsylvania. But obviously this slide is to show you that he is not alone. He's somewhere over here in this picture. But a whole team of people with diverse experiences is required to make such a thing happen. I'll end my talk by talking about the latest advances in gene therapy, and you all must have heard about gene editing technologies like CRISPR-Cas9. And the newer technologies where it involves non-coding RNA, where non-ATG RNA can generate proteins, and mucosal immunity is involved in this, has also been discovered. So new molecular biology is being discovered. And the next slide shows you even more dramatic events where new nucleotides are being synthesized. We know the 4 new nucleotides that we've known from our high school days, ACTG. Now, new nucleotides have been chemically synthesized, which are called X and Y nucleotides. And the first bacteria with 6-letter DNA has been made. These bacteria can be reproduced normally and have 6 nucleotides. So the field of molecular biology is exploding. So I'll end here by saying that I spoke a little bit about gene expression, targeting the gene expression, and also a little bit about gene editing. I'll end my talk by this quote from Jim Wilson. Gene therapy is a classic disruptive technology. It is so different, and it will impact the entire practice of medicine. But if we as a society, we as a society have to figure it out. We'll have some successes, we'll have some failures. It's still science. We are still learning, and this is not routine. And we are just at the beginning. So I'll stop here and also mention that there is a lot of things that we have to do as industry, academia, regulators to work on these concepts so that we can be successful. So thank you for your attention, and I'll be happy to take questions. On the topic of scientific writing, I've advised the professors that I will answer questions, and maybe we can do a special session on scientific writing. Thank you so much. Hello.
Hello.
Hi, Yogesh. Yes, yes. I'm happy to take. Yeah.
Hello. One question is: can COVID-19 stop the effect of a newborn baby by gene therapy or transplant? Like if COVID-19 newborn baby is there, can it be
Hello.
stopped by therapy or transplant?
Hello,
Yes.
sir, can you hear me?
Yes, I can hear you.
Sir, would you like to continue on the bit on the development of new biologics and your personal experiences and how you developed new biologics?
Of course, of course. Actually, it's been about a little bit more than half an hour. So I was not sure how long to continue the monologue. But I'll be happy to continue if people are interested in biologics. I spoke mainly on gene therapy. The biologics. If it is okay by everyone, I can do a biologics presentation separately because that will require a little bit more time. I speak mainly about gene therapy, and so I'll basically do it now or later. I'm okay either way.
So we can continue for more than 40 minutes.
Oh, you have 40 minutes more. Okay. Then why don't I do a little bit of presentation on biology? And we'll speak. So can you? I'll share my slides. Thank you. Now present. Okay.
So we can continue for more than 40 minutes.
Hello. Should I start the second part of the lecture, which is biotechnology? Hello. Should I start the second part of the lecture on biotechnology, or are there questions on gene therapy?
Hello. So we can take a few questions on gene therapy.
Okay. Yeah.
So our first question is there any treatment available for gene therapy?
Biotechnology, or are there questions on gene therapy? Please repeat the question.
So, can you hear me?
Yes, I can hear you.
Yeah. Can COVID-19 stop an effective newborn baby by gene therapy or transplant electropollate?
Yes. So this question is a good question. Multiple answers to this question. One is the gene therapy is being tried for treatment of COVID. For example, two or three methods are being used. So COVID has an RNA genome, right? So what is being done, what is being tried is what is called an antisense technology. Antisense nucleotides are being designed where gene therapy can be given to patients as a drug. So this antisense DNAs can go and bind to the RNA of the virus and block the viral replication. So that is one therapy for gene therapy for COVID that potentially can be used. The other is CRISPR-Cas9, which is the gene editing technology, where CRISPR technologies are used to cut the DNA or RNA of the virus and inactivate the virus in certain ways. CRISPR-Cas9 is also being used as diagnostics for detecting the RNA of the virus. So many ways the CRISPR-Cas9 is using. So now, as far as babies born to COVID-infected mothers and whether the virus is transmitted from mother to baby, I think that work is still very early. The disease is still a new disease relatively in humans. So it's difficult to predict what how babies will be infected by this particular disease. So right now, there is no treatment to prevent that right now. But I would imagine a vaccine in the mothers would prevent the virus from being transmitted from mother to child. So, thank you. I'll take the next question if there are any.
Okay, sir. Yeah. Sir, there is a multiple question on autism. Is there any gene therapy available for autism? If yes, where?
Yeah, good question. So, autism is a very complex disease. It's not a single gene defect. There are probably multiple genes. There are probably multiple environmental factors, social, many different factors that contribute to ultimately the treatment of autism, a gene like autism, a disease like autism. That is one of the reasons we started working, or people have started working on gene therapy with diseases like ornithine transcarbamylase, RPE deficiency. I showed you cystic fibrosis, or the hemophilia, for example, is because these are simpler genetic diseases because they are caused by a single gene defect. So if you correct the disease of the single gene, then you will get correction of the genetic disease. Diseases like autism and multiple sclerosis and other diseases where multiple genes are involved and we don't even know which genes are involved, the treatment for them becomes a little bit more difficult. Having said that, I'm not familiar with it, but there are people who are working on understanding the molecular pathogenesis of autism. Once we understand the molecular biology of autism, then the gene therapy will come into practice. I hope that answered the question.
Yes, sir. So there's one question. what are the ethical issues associated with gene therapy?
Very important. It is the ethics of gene therapy is a field of its own. You actually can get a master's and PhD in ethics of gene therapy in various universities in the world. The University of Pennsylvania, Arthur Kaplan, who's the pioneer in the field of gene therapy, ethics for gene therapy, has been doing ethics in gene therapy for many years. There are many components of ethics in gene therapy, starting from is it ethical to treat patients with gene therapy when you know that it is not going to work very well in the beginning? Two, who is eligible for getting gene therapy, and society has to make a lot of decisions. The last statement that I made from Jim Wilson's comment was, gene therapy is not a simple treatment of the disease. You're interfering with nature. So ethics is going to be an integral portion of gene therapy. And I'll tell you, for students, the field of ethics is a field of its own. It's become a whole, you can have your entire career just doing ethics work.
Sir, there's one more question. Has Luxturna as a drug for renal regeneration yielded 100% positive result in humans?
Well, if the question is related to the Luxturna, which was the eye disease, and there's no drug that is 100% efficacious. So there will be certain patients who will respond and certain patients who will not be responding. And that is the nature of all drugs. Even paracetamol. When you take paracetamol, a simple headache, for example, not everybody is, the drug is not effective for everybody, and this drug is 100 years old. So there is no drug that is 100% efficacious. The challenge for people who are developing drugs is to identify patients in which the drug will work the best. Those are called biomarkers. So that's the answer to that.
Okay, sir. So can carcinoma cure by gene therapy?
Yes. Again, gene therapy works very well if the molecular pathogenesis of the disease is known. If you know the molecular aspects of gene therapy, then the gene for that can be corrected. So like for the CAR T cell therapy that I presented for tumor, for leukemia, we know that CD19, which is expressed on leukemic B cells, is the target for killing these CD19 positive leukemic cells. And when we develop the CAR T cell, when the CAR T cell is developed, it is developed against the CD19 molecule. And so you can kill the CD19 expressing tumor cells. In the specific cancer that you asked, if you know the antigen, if you know which of the cells are involved in the tumor, then developing a target becomes a little bit easier. Yeah. Next question.
So gene therapy coupled with IVF to make subsequent changes in the character, is it possible?
It is definitely possible. In fact, it was in the news where a scientist from China deleted the gene for CCR5, which is the co-receptor for HIV, and put the stem cell back into the womb of a woman, and the children were born. So these children have no CCR5 gene expression, and it has created a whole ethical dilemma of whether this is the correct thing to do. It is, again, interfering with nature. This is a very important question that society will have to deal with. Technology, yes, you can do it.
So is there any gene therapy available for muscle dystrophy?
Yeah, so muscular dystrophy is one of those diseases where there are multiple gene defects, and many of these gene defects are slow progressing. So, for example, there's a gene called which is where the muscular dystrophy comes from. Dystrophin is a protein that is expressed on the membrane of the muscle cells, which allows the elasticity of the membrane of the muscle. So, for example, when we do any muscular activity, the muscle contracts and then expands. They have this contraction expression of a muscle. In the absence of this dystrophin gene, the muscle contraction expansion, the elasticity of the muscle is lost. So when it expands, like a weak rubber band, the muscle will not come back because this gene is absent. So now, because it cannot come back, the muscle cells don't work. So imagine the heart is a muscle. And so if the heart expands in these patients, the heart doesn't pump very well. So to correct this disease, even though we know the gene, we know that the dystrophin gene is affected, we will have to correct this gene at the embryonic level, at a very early stem cell level, because you can't treat a patient where every muscle fiber of the cell needs to be expressed with the gene therapy. So there are a few challenges in muscular dystrophy. Having said that, the latest gene therapy that is approved from Avexis and Novartis is a subset of a small subset of this muscle degenerative disease. Hopefully that answered that question.
Yes, sir, sure. Sir, do you know any institutes in India or the government agency which helps for gene therapy treatments?
As an institute, I don't think there is an institute for gene therapy, but there are many institutions that are doing research on gene therapy, and a lot of companies who are doing gene therapy research. Intas, for example, in Ahmedabad is doing some work. All the major institutions in India, from All India Institute of Medical Sciences, in the Department of Biotechnology, and even in the University of Mumbai, there are certain places which are doing various aspects of gene therapy. It's not just one gene therapy, because you have to understand viral vectors, you have to understand the molecular biology, you have to study the immune response to gene therapy, you have to treat patients. There are so many aspects of gene therapy. So a lot of these institutions are doing little work, and they all collaborate. The best would be to collaborate with everybody and do this work. Please. Good question.
Thank you, sir. Sir, there's one person who is saying, I have heard that gene therapies are more successful at earlier stages of life. As an individual ages, cautions related to the therapy increases. Is that true, sir?
Yeah. So many, again, let me see if I can answer the question in a logical way. If you think about diseases which are genetic diseases, let's take the example of ornithine transcarbamylase that I mentioned, which is the OTC gene defect, which is caused by this mutation in the OTC gene. This is a single gene defect. Now, when children are born with this disease, because they don't have this gene in their liver, they cannot convert the protein into urea. And because it doesn't convert to urea, because of the absence of this enzyme, ammonia increases and these children get brain disease, right? And they die of brain encephalopathy. To correct this disease, we have tried, the University of Pennsylvania trial tried to treat the disease by treating the liver with the adenovirus expressing OTC. You get very good expression of the OTC protein in the liver, but in the patient, what happens is the OTC gene, which is the normal OTC gene, is a foreign protein for that patient. The immune system thinks that the normal gene is foreign, almost like a virus, and rejects the foreign gene. So therefore, even if you have the best gene to correct the disease, immunology will still be a problem in treating diseases in adults or even in babies. So what gene therapy in these kind of genetic diseases needs to do is to start thinking about treating these diseases at the stem cell level. So if you can treat the stem cell, which undergoes immune education, then you will be able to treat the disease and avoid rejection. So this is an area, neonatal and prenatal gene therapy, that is being studied by many people around the world. And one of the pioneers his name is Alan Flake, and he's done a lot of work in this area.
So I think I will take a last question. There are many questions which I can mail you later on. The last question is, can Alzheimer be treated by gene therapy?
Yeah, good question. Again, my answer is the same as for autism. Alzheimer's is caused by multitude of genes, and when there are many genes, it is very difficult to treat, to understand how to treat these diseases. So, for at least for now, the gene therapy that we are talking about and the development of drugs for gene therapy involve treatment of those diseases for which we understand the molecular pathogenesis very well. If you understand the molecular pathogenesis of the disease, then it is easier to design the drug. Otherwise, it becomes very difficult to treat the disease if you don't know what the pathogenesis is. Yeah.
Thank you, sir. There are many questions which I can mail you later on. So if you want to take a 5 minutes break, you can take, and then we can continue with the next session.
Yeah. I'll be happy to do that. I would like a few minutes break, and then we can come back.
Okay, sure, sir. We can have 5 minutes break, and then we can continue.
Okay.
Yeah. So, can we start the session? Hello. Sir, unmute your mic.
Yes.
Yeah.
Okay. Maybe, should I share my screen?
Yeah, sir. You can share your screen, sir.
So now I'll speak a little bit about biotechnology and development of drugs. You let me know when I can start, and I'll start. Okay. So I'll get started.
Yes, sir. We can start.
Yeah. So again, thank you so much again for the time. This is amazing that we can talk about the second topic. I'll talk a little bit about biotechnology and how the field is changing, and I'll touch upon what you mentioned earlier. What is the role of academics? Today, in biologics, there's a lot of very good things that are happening. The way to mute everybody else is background noise. Hello? Yeah. Yeah.
So you can
Hello?
continue. Yes, sir.
There's a lot of background noise I can hear. That's why I just told Yogesh to probably mute everyone else. So India is big, very high youth percentage, high literacy rate. GDP was growing. I mean, obviously, COVID is going to tell us what the future is going to be, and we have a lot of potential in India. The biotechnology sector in the world is about almost $300 billion industry. In India, it's $11 billion. It's growing at a very, very rapid pace, 20% at CAGR, the majority of which is contributed by biopharmaceutical industry. The pace of discovery, and we talked a little bit about gene therapy earlier, since the sequencing of the human genome has been so rapid. It includes understanding of structural biology, protein expression systems, RNA interference, genomics and proteomics, pharmacogenomics, combined with the engineering sciences of informatics, has really empowered the field of biotechnology. The process of developing drugs starts in biologics, starts at the early level where a protein has to be produced. So biologics, unlike small molecules, has to be made by biological systems. Initially, the DNA usually for this protein is encoded and developed into a cell line, for which you need process and analytics to develop this process. Then you need pharmacology work to do. Then which is followed by toxicology and clinical trials. Then through the regulatory processes where you involve intellectual property, regulatory, business development, and operations, you finally get approval. So this whole process takes anywhere between 10 to 20 years for a biologic to be developed. So let's go through briefly of what these steps are during drug development. So the early process involves designing of the molecule. So you know what molecule it is, and you have to design it just like a car. You have to design a molecule. You develop a process and you determine what the variables are, and you do preclinical studies. And what is required in developing the molecule? In this case, I'm showing you an example, which is a drug. Here in this example, it's called Enbrel, which is a drug for rheumatoid arthritis. You have to take the gene for this protein and express it in a cell line. You can see the cells are growing this protein. You have to grow it in fermenters, which are shown here. And the fermenters then will produce the cells which will secrete this protein. This protein is purified through column chromatography. And then finally, it is packaged into this vial. So this is the whole process of what it takes to develop a biologic. It should be cost effective, it should be scalable, and it should be scientifically innovative. So all of these components are involved in a process. One of the drugs that is very popular in biologics is an antibody molecule. And why are antibodies so good as biologic drugs? Because they have high specificity in their FAB binding domain. They have a long half-life. There are some drugs where you dose only twice a week. They can be manufactured easily, but also they have some limitations because you need high doses for their production, and they have limited tissue distribution. So these are the biologic drugs that are being developed. So as you move from early preclinical stages, then you move on to phase 2 of the molecule, phase 1, phase 2, phase 3 of the clinical trials, at which time you have to optimize the process, define the process format, develop purification procedures to improve yield, robustness, and high concentrations of this drug. So a lot of work is done during the course of the 10 to 20 years until you get approval. This slide just shows a little bit about the specificity of monoclonal antibodies and how we can improve monoclonal antibody therapeutics by making toxins to the monoclonal antibody, making multi-specific antibodies, catabolic antibodies, and even siRNA. So coming back to the process of drug development, I showed you first that you need a process to develop the product. The next thing you have to do is toxicology. So what is the purpose of a toxicology study? The purpose is to establish the dose at which it can cause what is called dose-limiting toxicity, shown here in this red line. You give higher and higher doses of drug to monkeys or animals, and you determine at what dose is it now not safe. You look at safety. That is your upper bound of your dose-limiting toxicity. And you also have to look for efficacy in your pharmacology study. So the window between efficacy and toxicity is the window in which you can develop your drug. Pharmacology, also, you're trying to look for the efficacious dose, and you have established your dose-limiting toxicity. The y-axis is dose concentration, and the lower axis is now efficacy. When you increase the dose in an animal model of a disease that you're trying to treat, these are the elements of pharmacology that you have to study. The drug, when it is administered, is first absorbed, then it is starting to clear through the elimination phase, and then you have your terminal phase, and you have a half-life. And then at some point, there will be an efficacious dose. So the purpose of the pharmacology is to stay within these boundaries of efficacy and toxicity. The next generation of toxicology and all of these studies this is what it will take. From toxicology, the process will move from looking at toxicity to predictive toxicology using data analytics. For efficacy, we will have to do what are called adaptive clinical trials, and we'll have to use biomarkers to understand which patients are most likely to be impacted by the treatment that we are giving. Population PK/PD studies will have to be done. And then finally, adverse events and manufacturing also will have to transform in the field of biotechnology. So very exciting times for the field of biotechnology. Today, when drug development is approved, when a drug is approved in the market, you have to demonstrate that the drug is efficacious, you have to demonstrate that it is safe, and you have to have all the regulatory processes. But in the future, and now, we have not only to demonstrate that, you have to demonstrate what the value of the drug is for the patient, meaning nowadays the drugs are very expensive. It has to have some kind of a value associated with it. And then access. When the drug is available, many people don't get access to the drug. So we have to make it possible for people to get access to the drug. So these are the 5 things that are very, very critical in drug development. Today, in India, for example, there are a lot of different biologic drugs that are being approved in India. For example, erythropoietin, GCSF, growth hormone, streptokinase, interleukin-2, all of these are being made by multiple different companies. And these are some of the companies that are working on biologic drugs. For example, Dr. Reddy's, Biocon, Bharat Serum, MCure, Reliance. All of these companies are working on biologic drugs today. Nature Index is a scoring system in the magazine Nature, which scores how institutions around the world collaborate with each other. So if I just show you a map of India, there are a lot of these blue marks are the marks which are blue, yellow, and red marks are the marks where all the major institutions, biological institutions, are listed in India. This you can go to Google Maps and find this list, biotechnology in India, and find all the companies, all the academic institutions which are doing this work. It's a busy slide, but I want to quickly run this by you. When Nature Index did an analysis of the top 10 institutions in India versus top 10 institutions in the globe with respect to collaboration and doing innovative research, this is the scores that they have, and this is what it is in India, and this is from Nature. So you can see that the scores for the top global institutions are in 1400. For example, let's take this column. This is a score 1400 to 420, and a lot of different institutions in the world. And just look at the scores of what's happening in India. Yeah, so the score is quite low. And then these are the companies, relative score of the companies. So the collaboration report card for India and academia is quite low, right? And what we need to do is we need to demonstrate the biotechnology education, research systems, process of communication, and a multidisciplinary approach. I'm going to pause here because I'm not sure whether you are hearing me or not, and I want to confirm that before I move forward. I'm going to stop sharing for a second. Hello. It is very difficult for me to know whether you can hear or not.
Yes, sir, we are hearing you.
Okay, good.
Okay,
If someone can just keep saying yes or no, because I'm just looking at my slide and it's very difficult for me to know. Sorry about the interruption. But now let me move on to the important part of the presentation. It is going to be about careers.
Yes, sir, we are hearing you.
Yes, good. Yeah, I'm getting a phone call from somebody on the phone. I was wondering whether somebody from your group is calling.
We are hearing you.
Yeah, good. So I talked a little bit about the biotechnology and biology.
Yes, sir.
How it is improving. No, I think if you can actually now, I can just speak. So when I talk about careers, if you think about a career in biotechnology, you can see how many different careers you can have. Obviously, the science is the obvious career, like chemistry, biochemistry, molecular biology, and physiology. These are the kind of technologies that are required in pharmaceutical industry. But you also need chemical engineering to manufacture the product. You also need doctors and physicians in companies. You also need pharmacists. You need people to do mathematics and statistics. All of the data that is generated is in this area. You need computer science, engineering, production, mechanical engineering. You need all of these people to do work in the biotechnology sector. You also need management, marketing, regulatory, lawyers. You need all of these jobs in a biotechnology career. And you also need finance, economics, graphic art, medical writing. There are many, many careers in the field of biotechnology. So it's important for us to understand as educators that there are a lot of different areas that people can get jobs in the area of biotechnology. So let's compare what does the industry do and what does the academia do, right? So in academia, in a college or university, we have classrooms and we have curriculum, meaning you have a course, you have a syllabus, right? In industry, equivalent to that, there are scientific advisory boards. So, if you're working on a project, there'll be an advisory board who helps you develop what you're going to work on. In academia, there are short-term intensive projects. So students will come for a year, 2 years, 5 years, whatever the period is, and do a small project and write a report on the project. That is a deliverable. In industry, that has to translate into intellectual property. Yeah, that work has to relate in intellectual property. In academia, you have a lot of competitions, design competitions, and you do projects, and you have science projects and all that. In industry, you do marketing. You have to do marketing your science. You have to let people know what work you are doing. Then you in India, in academia, you have sponsored projects where you get grants from DPT and others, right? That's how you do your research. In industry, you need budgets. You need financial capital. You need venture capital to help you develop these drugs. And typically in academic institutions, you do presentations and conferences and all that for communication. In an industry, it is more of departmental collaboration. So, for example, the clinical department will talk to the manufacturing department, and the manufacturing department will talk to the toxicology department. So this is the kind of conversation that we need to have in industry. Yeah. So finally, the collaboration that involves education, training, science and technology, the micro and macro environment of the academic institutions will lead to innovation. The collaboration will lead to innovation. Yeah, and a lot of policies need to be developed to enable this innovation to happen. And finally, the most important aspect of all of this biotechnology and academia is this whole process of culture. You have to have high work ethics. You have to have a positive attitude. You have to have this resilience, ability to pursue, right? And you have to have all these other skills in addition to your science. You have to have decision-making skills, communication skills, and a high level of emotional intelligence. All of these make a full person, and that's what it takes to have a very positive career in your life. So I'll stop here, and maybe we can have a discussion on this.
Thank you, sir. Sir, can we have bit questions on these topics?
Absolutely.
So my first question is, which parameter defines pharmacokinetics property of novel biologics?
Yeah, good question. Now, in pharmacology, what you're trying to establish is you're trying to establish the minimum dose that is efficacious, right? So and when you do pharmacokinetic responses, it is by definition, when you give a dose to a patient initially during the time course, the concentration of the drug is very high. And then over a period of time, it reduces over time. So the parameters that are measured in pharmacokinetics are called ADME, which starts for ADME, absorption, distribution, metabolism, and elimination. So ADME are the 4 terms that are used for measuring pharmacokinetics. Yeah.
Okay. So while an animal trial stage is skipped in COVID-19 drug preparation?
Yeah. So, I don't think it's skipped. Probably a very small amount of work is done in that space. Everything in COVID is against all the rules that we are doing. Even clinical trials are being done much faster because of the urgency of what is required today. It is during drug development, it is always a combination of risk benefit. How much risk are you willing to take to get the benefit? Today, in the face of COVID, we really need drugs and vaccines and therapies which will have an impact. So we are willing to take the risk that the drug may not be safe, but as long as it cures COVID. So it's always this risk-benefit ratio, and that's why the speed is of importance today. There will always be risks. Yeah. Thank you.
So there is a very interesting question. Many youngsters are now, which are biotechnologists, they want to become an entrepreneur like you, and they want to start new companies. Does it require much of experience, or they should have good qualities or a knowledge, or they should have more experienced person in advisory board?
Excellent question. So let me first tell you, I am a very conservative entrepreneur. I started my company at the age of 58. I wish I had started it when I was 25 years old. I would have had a much different career path. I think the rules of entrepreneurship are not written in a book. Everyone has to try their own process. I think it's not an easy thing to do, but you have to have courage and determination. These are the factors that require entrepreneurship. A good idea and follow your dream. And whether it is opening a coffee shop or whether it is developing a drug, the kind of things that you need to do to become an entrepreneur are similar. So you have to be able to take risks, take investments. Obviously, you need a lot of advice. You need mentorship. You need a lot of that things. So if people are interested in this kind of a topic, I might do one session with you a little bit later in a couple of days, where we can talk a little bit more in detail about such topics. I'm very interested in talking about these. Yeah.
Sure, sir. Most of the students will be delighted because our government now is encouraging for young entrepreneur and startup companies. So students in our college also are motivated to do something similar to what you are doing, having a lot of experience and then becoming an entrepreneur, or starting an entrepreneur and becoming an experienced person in that.
Yeah. No, I think these are not simple questions to answer. Let's have a separate session on this. I think it's important.
So, what are the challenges which are coming across when you are formulating a drug? So for an example, if a contraceptive drug is formulated, what kind of challenges are faced in that?
Yeah, good question. The drug development process, like I showed you in the slides, has these specific steps that you have to go through. First, you have to design a molecule. Then you have to manufacture the molecule, make it in some way. Then you have to do pharmacology, which you have to find the efficacious dose at which it works. Then you do toxicology, and then you do clinical trials and see whether it works in humans. So there are distinct steps in the field of biotechnology or any drug development. So the challenges that are there will be there in every area. So for example, for the design, when you do a design and you want to look for a drug that is, let's say, a contraceptive that you asked for, let's say it's the drug is affecting some kind of a hormone that will affect your hormonal balances where you can regulate contraception. And you choose one hormone. Let's say that hormone is hormone X. That hormone X has 100 different drugs that can work on it. Now, the challenge will be, which of those 100 drugs will you choose to make it effective? It is not an easy thing to decide. So each step of drug development has many different challenges. And to go through these systematically, you will have to start learning about how every step of drug development works, first at the macro level, and then you can come down at the molecular level. Yeah. So, but if people are interested in understanding a little bit more about that, we can talk further in another session where I do a session on drug development.
So this is my question. Sir, if an academia has made a drug, I would say they have cloned a gene for a streptokinase or I would say tannase gene or any gene. Can they approach any industry for doing a clinical trial, or a company can pick up that drug?
Yes, absolutely. You should be able to do it, and that is the part of the collaboration that I talked about, academia and industry. And it is important to have these conversations with industry teams, and you need an industry mentor, for example, someone who has worked in the industry to develop the drug. So you have to first find a company which may be interested in the area of work that you are interested in. Like for example, Biocon, I work for Biocon, and if you're working on, let's say, Alzheimer's disease, let's say you've got a drug for Alzheimer's disease, it is unlikely Biocon will work on Alzheimer's disease because that's not what they work on. They work on diseases like cancer and diabetes. Just giving an example. So if you want to work, if you have a drug, you have to go and collaborate with a company that works on Alzheimer's. There, the likelihood of them working with you is higher. So you have to do some background research to see which company is the best company to work with you for. So those kind of things need to be taken into consideration as you decide to do a collaboration. Not simple. Yeah, please.
So there is one question: what are the challenges faced during the analysis of biologics?
Yeah. Analytics of biologics and any drug development is actually very, very important. It is the eyes and ears of the drug development process. So when you do analysis, you're actually taking a molecule and trying to see how it looks and how it feels and all the structural elements. So when you look at the structure of a molecule, you need a whole degree of analytics to measure all kinds of things in that molecule. You look at the primary structure, the secondary structure, the tertiary structure, and the quaternary structure of molecules. So for all of these, you need very, very sophisticated technologies. So sometimes access to technologies like cryo-EM is not there. There's only one cryo-EM in all of India. And so how do you get access to that technology to be able to understand the structure of your molecule? That's an example of an analytical. Many, many challenges.
Okay. Thank you, sir. Sir, there are more questions which I can mail you later, and then I can eventually mail the participants accordingly. Sir, would you like to just a brief 5-minute session? What should be the key for doing a scientific writing?
No.
And we can have that session later on, sir.
Yes, there are a lot of aspects of scientific writing, but I think it might require a special session. Like I said, there might be a session that we might want to do. We can plan systematically.
Hello, sir.
Yeah. Hello.
Yeah. Can we have, sir, one last question?
Absolutely.
So what is the difference between pharmacokinetics and pharmacogenomics?
Okay. Pharmacokinetics is the study of the drug in the body. So when you inject the drug into the body, you look at the ability of you measure the drug and look at the kinetics of the response in the patient. Pharmacogenomics is the ability of the drug to interact with your genome. If the drug is interacting with the aspect of your of your molecular biology, you're looking at that interaction, and then also how genes are being upregulated and unregulated during the treatment of the drug. That is the pharmacogenomics.
Hello.
Right.
Hello. So we can't hear you.
Yeah. So I mentioned the difference between pharmacokinetics and pharmacogenomics.
Okay. Yeah. Sir, I think we'll end the session here only. And it was, I would say, William Author had said very gratefully, a feeling of gratitude and not expressing it is like wrapping a present and not giving it. Sir, by your lecture was most wonderful gift wrapped in the ocean of knowledge and presented to us. For this, our college, Guru Nanak Khalsa College and JNID department would like to thank you for your time and lecture, and we'll like to have more sessions with you in future. Hope this lockdown helps to cut off and we can have a very live session with you in person and take you that opportunity. I would also like to extend my thanks to my team Yogesh and Raza, our technical team Aldrin, Sham and Ashish for their support. My appreciation would not be fulfilled if I don't appreciate my senior mentor who encouraged and supported me in all the times, Dr. Sukhjeet Gujral. Sir, from our heart, I really want to thank you for your time and lecture. Thank you, sir, so much.
Thank you very much. Thank you so much.
Yeah.
Bye-bye.
Dear participants, we'll be sharing the feedback link in the YouTube and we also be sharing through the Telegram. You can connect it and you can get your certificates between 20 to 25 days. Okay?
Thank you.
Yeah. Thank you, sir.